Thank you. It's a great pleasure for me to be here to present our research result concerning the potential application of nanomaterial for crop nutrition. This research was a 3-year PhD program conducted by Christophe Trouillefou and supervised by Claude Plassard and Emmanuel Bellamy. This research program was funded by the SupAgro company, part of the Rookie Group, and involved 2 laboratories. One specialized in nanochemistry, and the other one more focused on pea nutrition. Therefore, it can be considered as a multidisciplinary approach for the use of soil phosphorus for crop nutrition. I want to start this talk by sharing with you my fascination for nanoscale studies. Nanoscale is very important because it is at that scale that atoms and molecules interact together and assemble into unique structures which have specific properties. That's why we focus in this talk about nanomaterials. Briefly, what is you have on the left part of the slide a general pattern of synthesis of those nanomaterials, and I focus on silica-based nanomaterials. From a surfactant, a template is generated, and then silica is polymerized with this template. This template is then removed, for example, by calcination, and you obtain those nanoparticles which have specific porosity and structure. If the internal porosity or diameter is between 50 and 2 nanometers, the materials are qualified as mesoporous. And in this talk, we are interested in silica-based mesoporous nanomaterials. During the synthesis, you can control a wide range of properties. And these properties are listed here. The most important that you have functionalizable surface sites, and you can control the pore side and the structure. The materials can have a structure which is ordered or not. And due to those specific properties, you can have an effective protection of enzymes which are immobilized inside the porosity. And the protection is very important for temperature, for pH, but also for when there is presence of inhibitors. And moreover, as you can control the porosity and the connectivity between pores, you can manipulate mass transport of substrate and products. That's why those materials are widely used in medicine or water treatments in many application fields, and they are used for biosensor, biocatalysis, and so on. But until now, there is few development of those nanomaterials in plant nutrition. Some examples are cited for pesticide application. So in the context of resource scarcity and phosphorus needs phosphorus resource to meet people needs, we should consider the application of nanomaterials as another alternative for crop nutrition that we have discussed in this workshop. And we focus on phytate because phytate is an important source of phosphorus. But its use requires manipulating efficient extracellular soil enzymes, phytase, and those critical points are listed here. If we want to add exogenous phytase, we have to avoid binding onto soil minerals and organic matter. We have to stabilize the enzyme against pH and temperature changes in soil, and we have to avoid proteolysis that occur in soil. So our hypothesis was, can we use nanomaterials to stabilize phytase enzyme? So we use the Aspergillus niger phytase, which is a recombinant enzyme produced in a yeast Pichia pastoris. And we use this nanomaterial, which name is Grit6, this is the name, And this nanomaterial is another mesoporous silica base. You have here the complete description of the synthesis. Please pay attention that the synthesis requires energy for, during calcination and the use of non-friendly environmental precursor. I'll be back to this in a few minutes. And for the immobilization method, we tested a lot of immobilization methods, and finally we selected the simplest one, which is adsorption, which is a physics option. On which to immobilize the enzyme, we simply mix the extract of Aspergillus niger with a nanomaterial with appropriate condition of pH and ionic strength. And after washing the enzymes, we have, after washing the loosely bound or unbound enzyme, we obtain nanomaterials in a directly usable form. Here you have some characteristics of the enzyme, and you may remark on the main axis here, you have the main length of the enzyme. And here you have a picture, you have a microscopic picture of the kit 6, and you can see the order structure of this material. And here you have the internal diameter of the material, which is 11 nanometers, which is so larger than the enzyme. For a full description of the characteristics, the Erics and so on, there is a publication associated to this talk. So we focus more about the potential of this material. So we measure the phytase activity versus free enzymes. So we measure pH and temperature stability. We measure if there is a protection against proteolysis. And we use a control, which is another kind of nanomaterial, which have tiny pores, so the phytase cannot penetrate into the porosity. And finally, we use those immobilized enzymes to know if the phytase activity is enough to permit plant growth. So you have the condition here of the plant experiment. We use phytate as a sole source of phosphorus. We only use sand, and we have sterile conditions to avoid any contamination of microbes who can produce also phytase. The control was an absence of mesoporous, and we measured the P accumulation in plant tissue over 30 days. So my first result is this one. You may remark that the free phytase activity is much more than the immobilized activity, so I hope you are not too disappointed. I have later results that will be more, you will be more interested in those results. But you can see that the kit 6 have a phytase activity, which is more important than the control, because in the kit 6 you can have phytase, which is adsorbed onto the surface, but also phytase can penetrate into the porosity. That's why we have a greater phytase activity compared to the control. Despite the severe drawback of a lower phytase activity, we have a greater stability of the enzyme. After exposure to 55 degrees, the phytase activity dramatically decreases, whereas the kit 6 phytase remains remarkably stable whatever the exposure time of the enzymes to 55 degrees. And when we consider now the main characteristic of the phytase, you can see that you have here the phytase activity, and here you have a range of temperature. You have here the free phytase, and you can see its optimum, its second optimum, in fact. And here you have the optimum of the immobilized enzyme. You can see that there is a shift of the optimal pH, but there is the decrease of phytase activity in the immobilized enzyme is less sharp than the free phytase. And for the pH, we have also the same result. if we consider now the remaining activity after a trypsin treatment, so trypsin is a protein which can lyse, yes, we can lyse the phytase. You have different phytase loadings in the nanomaterials, and after a 5-minute trypsin, we have a decrease, of course, of the phytase activity, mainly due to the lysis of the phytase at the surface of the nanomaterials. But trypsin is a small protein which can penetrate into the porosity, so the phytase inside the pore is also degraded. But due to the connectivity and to the penetration of the phytase, there is still a remain active phytase activity, which is mostly the same for the different loading of enzyme. So the hypothesis that there is two kinds of phytase adsorption in the nanomaterial is confirmed by this result. Here you have the experimental data, and here you have the modelization, the model of a long Mir-Freundlich isotherm, and you can see that here you have a difference between the model and the experimental data, and this part is probably due to the external phytase. And this is the same for the control, of course, but there is less phytase. So there is a multi-layer adsorption at the surface of the nanomaterial. Yes. So the main result, which is, I guess, the most important in of this talk, is the measure of the P accumulation in plants. This is the control, remember, no mesoporous, and this is plants which were grown in presence of KIT6. And we can see that plants can accumulate much more phosphorus over 30 days compared to controls in sterile conditions with phytate as a sole source of phosphorus. And this is the same for shoot and roots. So in conclusion, nanomaterials are really promising, yes, but remember, KIT6 has a high carbon footprint, and its precursors are not really eco-friendly. So we tested an alternative, an encapsulate enzyme into phospholipid. So it's a direct encapsulation in one port encapsulation with different phytase, one from Aspergillus niger, one from Bacillus subtilis, and you can see it's a very promising result because we measure P liberation rate, which are very important. So my last slide, nanotechnology is no longer sci-fi in agriculture. Those materials are promising, but still in infancy to use phosphorus. And maybe those nanomaterials can also be used as biosensors of phosphorus dynamics in soils. Thank you for your attention.